Hello everyone, welcome to BioQuick. Today our topic is glycolysis. This is a very important topic. There are 10 steps in glycolysis and you need to know the substrates, the products and the enzymes which are used in each steps. So, first, what is glycolysis? Glycolysis is the pathway by which glucose is converted via fructose 1,6-bisphosphate to pyruvate with the generation of 2 moles of ATP per mole of glucose. It means that in glycolysis, one molecule of glucose will give 2 moles of ATP and 2 moles of pyruvate. Now, let us see. Glycolysis is also known as EMP pathway. What is the full form of EMP pathway? E means Emden, M means Meyerhoff, and P means Parnas. Glycolysis is a 10-step reaction which can be divided into two parts. First, 1 to 5 steps. It is known as the energy consuming phase or the preparatory phase or the glucose activation phase. And the second is from step number 6 to step number 10. It is the energy conserving phase or the payoff phase, that is ATP produced. These things are very important for NEET purpose. This is the total pathway of glycolysis. So here we can see glucose is converted to glucose 6-phosphate. The enzyme used is hexokinase. Here ATP is required and ADP is released. The second step is glucose 6-phosphate to fructose 6-phosphate. The enzyme required is phosphoglucose isomerase. Third step is fructose 6-phosphate to fructose 1,6-bisphosphate. ATP is again required and ADP is released. The enzyme used is phosphofructokinase. Number 4 is fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and dehydroxyacetone phosphate. It is known as DHAP and this is known as GAP. Now, step number 4 is fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and dehydroxyacetone phosphate. The enzyme required is aldolase. Here, the dehydroxyacetone phosphate is transferred to glyceraldehyde 3-phosphate. Here, one enzyme is required. It is known as the triose phosphate isomerase. And here, then glyceraldehyde 3-phosphate is then changed to 1,3-bisphosphoglycerate, and the enzyme used is triose phosphate dehydrogenase. 2 NAD plus is required and 2 NADH plus H2 is released. Step number 7 is 1,3-bisphosphoglycerate is changed to 3-phosphoglycerate and the enzyme used here is phosphoglycerokinase. Here 2 ADP is required and 2 ATP is released. Then 3-phosphoglycerate is changed to 2-phosphoglycerate. The enzyme required is phosphoglyceromutase. The ninth step is 2-phosphoglycerate to phosphoenol pyruvate, which is also known as PEP. Here enolase is the enzyme and 2H2 is released. The fifth step is phosphoenol pyruvate is converted to pyruvate and 2 ADP is required and 2 ATP is released and the enzyme used here is pyruvate kinase. Now we will go step by step and we'll understand every step and the use of each enzymes. The first step is hexokinase. It is the first ATP utilization step because first of all it is the first step and ATP is required. That's why first ATP utilization. And the enzyme here glucose is converted to glucose 6-phosphate. The enzyme used is hexokinase. Hexokinase belongs to a group of enzyme which is known as kinase. Its work is to transfer a phosphoryl group between ATP and a metabolite. Here the metabolite is glucose. It transfers the phosphate group from ATP to glucose. Here we can see in glucose 6 phosphate, in C6 phosphate, here we can see a phosphate group. That means a phosphate group has been transferred from ATP to glucose. That's why glucose 6 phosphate is formed. And Mg2 plus is the ion which is required. And the products are glucose 6 phosphate, ADP, and AH plus is released. This is step number 1. Step number 2, phosphoglucose isomerase. Here, glucose 6 phosphate is converted to fructose 6 phosphate. The enzyme required is phosphoglucoisomerase. Why isomerase group is used? Because there is just a isomerization. Isomerization is happening. Isomerization changes the substrate to their corresponding isomers by intramolecular rearrangement. Here we can see that an intramolecular rearrangement happens. That's why isomerase group of enzyme is used. Here, this is step number 2. Glucose 6 phosphate is changed to fructose 6 phosphate. Step number 3. It is the second ATP utilization. Why second ATP utilization? Because in step number 1, it was the first ATP utilization, and here also we require ATP, but it is the second time we require ATP, so it is known as the second ATP utilization. Here fructose 6 phosphate is changed to fructose 1,6 bisphosphate. Here the enzyme used is phosphofructokinase. Again we see kinase here. That means a incorporation or a movement of phosphoryl group from ATP to fructose 6 phosphate, and thus here fructose 1,6 bisphosphate is formed along with ADP and AH plus. This is step number 3. Step number 4 is aldolase. Why aldolase? The enzyme used here is aldolase. Fructose 1,6 bisphosphate is broken down into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and the enzyme used here is aldolase. Here aldol condensation is taking place, that is breaking of fructose 1,6 bisphosphate into two parts. Here step number 5, triose phosphate isomerase. The enzyme required in this step is in this reaction is triose phosphate isomerase. Here isomerization takes place. Isomerization between dihydroxyacetone phosphate, which is a ketose. It converts to a aldose, which is glyceraldehyde 3-phosphate. And the intermediate here is enidiol intermediate. Here we can see that that C2 carbon, it consists only a COO group, and here in glyceraldehyde 3-phosphate it consists of a OH group, and in C1 carbon it becomes COO. Thus there is a isomerization happening. What is isomerization? There is a intramolecular rearrangement taking place. This is step number 5. From step number 5, we get to know that two glyceraldehyde 3-phosphate molecules are formed. One is from step number 4. In step number 4, one glyceraldehyde 3-phosphate is formed, and again in step number 5 we see that dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. Thus two molecules of glyceraldehyde 3-phosphate are formed. Step number 6, glyceraldehyde 3-phosphate dehydrogenase first high energy intermediate. Here glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate, or which is known as 1,3-BPG. Here we require NAD plus and an inorganic phosphate. The enzyme used here is glyceraldehyde 3-phosphate dehydrogenase, which is also known as GAPDH, and the products are 1,3-bisphosphoglycerate and NADH and H plus. Dehydrogenase, it means that there will be a dehydrogenation, that is removal of hydrogen. Here we can see in C1 carbon, in glyceraldehyde 3-phosphate there was a hydrogen, but in 1,3-bisphosphoglycerate there the hydrogen has been removed and phosphate group has taken its place. Thus the name of the enzyme is dehydrogenase. This is step number 6. Step number 7 is phosphoglycerate kinase, first ATP generation. It is the first step in ATP generation. In this 1,3-bisphosphoglycerate to 1,3-bisphosphoglycerate, ADP is added and 3-phosphoglycerate is formed and ATP is formed. And the enzyme required is phosphoglycerate kinase. See, phosphoglycerate kinase. Kinase means there will be a transfer of phosphoryl group between ATP and the metabolite. Here the metabolite gives its phosphate group to ADP and thus ATP is formed and 3-phosphoglycerate is formed. Here in C1 carbon we can see that there has been a removal of phosphate group. This is step number 7. It is a very important step because it is the first ATP generation step. Step number 8 is phosphoglycerate mutase. Here 3-phosphoglycerate is converted to 2-phosphoglycerate. The enzyme used here is phosphoglycerate mutase. Mutase belongs to a isomerase group of enzyme in which there is a transfer of phosphoryl group. Here we can see in 3-phosphoglycerate, in the phosphoryl group is in the C3 position, but in 2-phosphoglycerate the phosphoryl group is in the C2 position. Thus there has been an intramolecular change in between these two these two metabolites, and the enzyme used is that's why the enzyme used is phosphoglycerate mutase. The pathway of synthesis of 2,3-BPG in erythrocyte is a detour from the glycolytic pathway. Here 1,3-bisphosphoglycerate is changed to 2,3-bisphosphoglycerate or 2,3-BPG. The enzyme used here is bisphosphoglycerate mutase. Here it again intramolecular change in the position of phosphate group occurs, and then it is changed to 3-phosphoglycerate. The enzyme used here is 2,3-bisphosphoglycerate phosphatase, and inorganic phosphate is released. And now this 3-phosphoglycerate returns to the glycolytic pathway and it changes to 2-phosphoglycerate, which we have seen in the last step. It is step number 8. In this 3-PG changes to 2-PG. Now step number 9. It is the second high energy intermediate formation. 2-PG or 2-phosphoglycerate is changed to phosphoenol pyruvate or PEP, and H2 or water is released. The enzyme used here is enolase. Here we can see that hydrogen group of C2 carbon and OH group of C3 carbon becomes H2O, which is this one, and here a double bond is formed, and that's why the name of the enzyme is enolase, and it is for enol pyruvate is formed. This is step number 9. Step number 10 is pyruvate kinase second ATP generation. The first ATP generation was in step number 7, where we have used the change from 1,3-bisphosphoglycerate to 3-phosphoglycerate. There we have seen it is the first ATP generation, and step number 10 is the second ATP generation. Phosphoenol pyruvate or PEP is converted to pyruvate. Here ADP is used and H plus. The enzyme used here is pyruvate kinase. Kinase means removal or the transfer of phosphoryl group between ATP and the metabolite. Here we can see that the metabolite transfers its phosphoryl group to ADP, and hence ATP is formed and pyruvate is also formed. This is step number 10. And now we have some points to remember which are very important. Step number 1, step 3 and step 10 are the rate determining step as they are irreversible in nature. So in the whole there are 10 steps. Among them only 3 steps are irreversible, and thus they are the rate determining steps. The second point is total number of ATP produced is 2. Second point is total number of ATP produced is 2. Why? 2 ATP is required in the first and third step. We have seen in the first and third step we have to give 2 ATPs, and 2 ATP is released in the seventh step. In the seventh step we have seen 2 ATP is released, and 2 ATP is released in the 10th step, and thus the total ATP is 4. But as we are using 2 ATP, so you have to deduct 2 from this 4, and thus the total number of ATP produced is 2. The third point is total number of pyruvate formed is 2. In this slide we see that in glycolysis what is formed? ATP is formed, carbon dioxide is not formed, water is formed. So in under ATP, in substrate level phosphorylation 4 ATP is formed. In ETS 6 ATP is formed. So in total 10 ATP is formed. We use 2 ATP. So thus the net gain is 8 ATP. This thing is very important. These calculation of the number of ATP are very important for MCQ. The formation of H2O. Directly we get 2 H2O. Indirectly, that means by ETS we get 2 H2O. That means the total number of H2O formed is 4. But we use 2 H2O molecules, and thus the net gain in H2O molecule is 2. These two calculation of ATP and H2O are very important. Now we come to conclusion, and we again just go through the go through the cycle. Glucose changes to glucose 6-phosphate. Here it is the first step. ATP is required and ADP is released. Hexokinase is the enzyme. Glucose 6-phosphate changes to fructose 6-phosphate. Phosphoglucoisomerase is the enzyme. Fructose 6-phosphate changes to fructose 1,6-bisphosphate. ATP is again used and ADP is released. The enzyme used here is phosphofructokinase. Step number 4 is fructose 1,6-bisphosphate. It changes to glyceraldehyde 3-phosphate and dehydroxyacetone phosphate. And the enzyme used here is aldolase. Dehydroxyacetone phosphate changes to glyceraldehyde 3-phosphate by using the enzyme triose phosphate isomerase. The next step, glyceraldehyde. Thus, from here we get 2 molecules of GAP. Then glyceraldehyde 3-phosphate changes to 1,3-bisphosphoglycerate. Here NAD plus molecules are required, and NADH and H2 is released. The enzyme used here is glyceraldehyde 3-phosphate dehydrogenase. 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. Here ADP is given and ATP is released. Enzyme used here is phosphoglycerokinase. The 8th step is 3-phosphoglycerate is converted to 2-phosphoglycerate. The enzyme required is phosphoglyceromutase. The 9th step is 2-phosphoglycerate is converted to phosphoenol pyruvate. Enolase is the enzyme and H2O is released. 2 molecules of H2O is released, and then Phosphoenol pyruvate is converted to pyruvate. The enzyme required is pyruvate kinase, and 2 ADP molecules are given and 2 ATP molecules are released. So this is all about glycolysis. If you like our video, then please click the like button and do subscribe. Thank you.